Our study was conducted by Mark Brandon, Julie Koenig, Jill Lloyd-Gibb, and myself. Our laboratory studies circuits for spatial processing and which give us a sense for where we are in the environment. These circuits overlap substantially with those for memory processing, perhaps because both functions require that multiple modalities are integrated. Unraveling how the brain can generate a sense of space began with the remarkable discovery of place cells in the rodent hippocampus by John O'Keefe. Ever since these place cells were discovered, one of the key questions was how this code is generated from pulling together different types of information. A major breakthrough in addressing this question was the discovery of an even more spectacular cell type upstream of the hippocampus by Hafting et al. In the Moser laboratory. Grid cells fire in multiple locations, which remarkably are arranged in a hexagonal grid pattern. These cells are most abundant in the superficial layers of entorhinal cortex, which provide the major input to the hippocampus. An obvious model is that grid cells provide the hippocampus with a spatial signal that is necessary to generate place cell firing. To test this model, we were able to take advantage of our finding that grid cells no longer fire in a spatial pattern while one of the subcortical inputs to the entorhinal cortex, the medial septal area, is inactivated. We have previously shown that place cells in familiar rooms are nonetheless retained without grid cell input. Theories on memory coding suggest that partial inputs are needed to remember maps, but that more complete inputs are required to first generate the map. The next big question was therefore, could the hippocampus generate a new map of an environment that had never been visited before without a spatial signal from grid cells? Here with me today is Mark to present the study.
To examine the question of whether the hippocampus could generate a new spatial representation without grid cells, we performed mu-small inactivation of the medial septum as animals navigated in a familiar and a novel environment. First, we found that inactivation of the medial septum caused roughly an 80% reduction in the power of theta oscillations in the hippocampus and a reduction in the spatial firing properties of grid cells in a familiar and in a novel environment. The key question then was, during this period in the novel environment, could the hippocampus generate spatial firing patterns when the medial septum was inactivated? And to our surprise, what we found was that spatial firing patterns emerged almost instantaneously as the animals were brought into the new environment and remained stable across subsequent exposures to this same environment. These newly formed place fields were very similar in size and stability as those that were formed after control infusions into the medial septum. Next, we asked if the spatial representation that was generated in the novel room was similar to the spatial representation that was previously established in the familiar room. And what we found is that these two spatial representations were completely orthogonal or different from each other, suggesting that even without grid cell inputs from the medial entorhinal cortex, the hippocampus was able to generate a distinct new representation of this novel room. Finally, we asked, even though place fields can emerge without grid cells, perhaps grid cells may gain control over these fields when the medial septum inactivation wore off. To examine this, we obtained recordings from the same neurons 24 hours after the mu-small inactivation when mu-small was metabolized and observed the firing locations of these fields. We found that the spatial firing patterns of these fields was maintained and stable even though grid cells had come back online. This suggests that grid cells did not change or control the spatial firing patterns of place cells when grid cells came back online.
In summary, these results challenge models that suggest that the information flow is exclusively feed-forward from grid cells to place cells and suggest that there's other inputs that can be predominant.